Today we will be talking about comparing solutions, particularly solutions with different osmotic pressures. You may remember that osmotic pressure is the tendency of water to move into a solution. Today we'll be focusing on isotonic, hypertonic, and hypotonic solutions. In an isotonic solution, they both have the same osmotic pressure, both solutions being compared. So if we have a red blood cell in a solution, that means that there is the same osmotic pressure of solution going into the cell, the water, as going out of the cell, because there's the same number of particles in the cell and outside the cell. If we were to do this in a tube in a lab, it would look something like this. We put a semi-permeable membrane right here, and both sides would be equal because there would be the same number of dissolved particles on each side. So the amount of water flowing this way is the same as the amount flowing this way. In a hypertonic solution, water will tend to flow into the solution. So if we have a red blood cell in a hypertonic solution, so the solution surrounding the red blood cell is hypertonic, the cell is going to shrivel up and look kind of funky because all the water will be leaving the cell to go into the solution, because the solution has more particles in it, and the water wants to go to where the most particles are because it wants to make an even amount of water in everything. So the water is going to leave the cell and it's going to shrivel up the cell. If we were to do this in a tube in a lab with a semi-permeable membrane, it would look something like this if there was fewer particles over here than over here. The net osmotic pressure would be going this way, so the water would be traveling, in this case, to the right. In a hypotonic solution, the water will tend to flow out of the solution. If we have a red blood cell, the water is flowing out of the solution surrounding the cell and into the cell because there's more osmotic pressure pulling it into the cell, which a lot of times will cause a red blood cell to burst. This is why it's so important to drink things like Gatorade and not just water, because if you have more particles, more density inside your cells than outside, it can cause them to burst because the water goes into the cell. If we have a U-tube in a lab with a semi-permeable membrane, and this side is our hypotonic solution, and then the water is going to flow out of it and into the other one. So this is our hypotonic side. So it's going to flow out and into this one because this one will have more particles in it. As you can see, hypotonic and hypertonic are just comparing each other. In a hypertonic is the one that has more of the particles floating around in it, so the water is going to go towards it. The osmotic pressure pushes the water into a hypertonic solution because it wants to balance out the particles in the amount of water. And in a hypotonic solution, it's leaving that solution, it's going out. So these are all just different ways to compare solutions. You're usually just comparing two solutions. In isotonic, they're the same. Hypertonic, it's flowing out of one solution and into your hypertonic solution. And in hypotonic, it's flowing out of the hypotonic solution and into your other solution.